Hi everyone, welcome back from the break. Hope everyone had a good holiday, and yeah, it feels like it's been a long time, which I guess it has been. It's been about 2 weeks. So we'll do 2 hours today. Hopefully it doesn't take all 2 hours, but we will cover the last 6 or 7 lectures, I think. So you should have 2 worksheets. They're just there on the table if you haven't got one already. Okay, so we'll get started on tutorial 6. So I just want to go over the central dogma. So a dogma is something that's like always correct. And then when you talk about the central dogma in cell biology, it describes the flow of information inside of your cells. Okay, so the flow of information, and that's going from DNA to RNA to proteins. Okay, so when you go from DNA to RNA, that's transcription, and going from RNA to protein is translation. And then each piece of DNA that codes for a functional product is called a gene. Okay. And so the gene product is not necessarily always a protein. It can sometimes be an RNA as well. So for example, transfer RNA or tRNA, that's the functional product. So it doesn't always have to go from RNA to protein to become a functional product. But most of the time it's going to be a protein, that end product. And then if you want to create DNA from RNA, that's going to be reverse transcription because you're going the opposite way. Okay, and you have retroviruses. So retro means backwards. So retroviruses such as HIV, they use reverse transcription. And we can also use reverse transcription when we're wanting to make recombinant proteins as well. And so if a gene is expressed, it means that the gene has been transcribed into mRNA and then translated into a protein. Okay, just because I think a few people get confused on what gene expression actually means. So the expression of a gene is talking about whether it's getting actually turned into a protein or not, or whether it's just sitting there in the cell. Okay. So next page. Then we've got the regulation of gene expression now, and that's going to involve transcription factors. So every single cell inside your body contains the same DNA, and that means that every single cell in your body, whether it's your eye cell, your brain cell, a bone cell, they have the coding or the DNA for every single gene, which in turn will create every single protein in your body. But you all know that your brain cell, for example, doesn't produce the protein that a bone cell would produce, right? So we need to be able to control the gene expression in every cell. And the way we do that is by having things called transcription factors. Okay? So each gene contains a regulatory region, and sometimes it's called a promoter region. So when they say regulatory or promoter region, it pretty much means the same thing. Some people will say that the regulatory region contains the promoter region, but I think for BIOC192 you can get away with just saying that either regulatory or promoter region. Okay, so that's going to be this promoter region is going to be upstream of the the DNA sequence that's going to be transcribed. So when I say it's upstream, you have the gene here. So this is DNA. And let's say this part here is the part that gets transcribed, and then this part here is the promoter region. Okay, so this promoter region is where the protein RNA polymerase, so this thing here is RNA polymerase. Okay, so it's got A's at the end of the name, which means that it's an enzyme. Okay, and you can kind of tell what it's doing by looking at the name. So it's an enzyme that creates a polymer of RNA. A polymer is just lots of things put together. Okay, so it's creating that mRNA transcript, and it's going to, this RNA polymerase is going to move this way. So this way is downstream, and then so the opposite way, the opposite way would be upstream. So like running, so like trying to swim against the current in a river. Okay, and the promoter region is always going to be upstream of the part that's going to be transcribed. Okay, and this is the promoter region is where RNA polymerase binds, and also other transcription factors like activators. You can have activators binding, or you could have repressors binding. Okay, so transcription activators will increase gene expression, while repressors decrease gene expression. Okay, and on this topic you were given two examples of cells, and that was the pancreatic beta cell and the liver cell. So pancreatic beta cells, they control blood glucose levels. So in order to control the blood glucose levels or blood sugar levels, they have to produce insulin. So they're going to express the gene for insulin. And remember that all of the cells inside your body have the gene for insulin, but it's only the pancreatic beta cells that will actually express the gene. Okay. But the liver cell doesn't store glycogen. Oh, sorry, that should say, however, since the pancreatic beta cell does not store glycogen, the pancreatic beta cell doesn't express the gene for glycogen synthase. So the glycogen synthase gene obviously creates the protein that will synthesize glycogen. So if you don't need to store glycogen, then you don't need to express that gene. Whereas liver cells, they are responsible for maintaining a store of glycogen as a source of energy in between meals. Therefore, they express the gene for glycogen synthase, but not insulin. Okay, so you'll come across glycogen a bit more later on in BIOC. Okay, and whether these cells express the genes or not will be determined by the presence or absence of the transcription factors. Okay. And in real life, there's actually like a lot more complex mechanisms that control gene expression, like methylation of DNA and like acetylation of the histone proteins. But unless you go into like more like second year level stuff, you don't really need to know that stuff. So then we'll get into dominant and recessive mutations. So every cell in your body contains 2 copies of the gene, not just one. You're going to have a paternal copy and a maternal copy. So paternal is from your father, but maternal is from your mother. And usually they're going to be exactly the same, right? They're going to have the same nucleotide sequence, which means that the protein that's produced will be exactly the same, or they could be slightly different. And if they're slightly different, they're called alleles. The different versions are called alleles. And both of these alleles are going to be transcribed in the cell, so that half of the protein that's in your cell is going to be created from the maternal allele, and half of the protein is created from the paternal allele. So I'll just draw that out for you guys, just so you can see what I'm talking about. So here's your normal cell, and here is so the cell is going to contain a nucleus, okay, and inside that nucleus you're going to have 1 copy of the gene. So let's say this is your paternal allele, okay, and then you're also going to have a maternal allele, okay, and these are both coding for the same gene or the same protein. Now they're both going to be transcribed. So transcription, and you've got the ribosome. So remember, the ribosome is the piece of cell machinery that's going to process the mRNA. So you've got so this here is your paternal mRNA, which has come from the paternal allele. And you've got, this is your maternal mRNA, which has come from the maternal allele. And then they're both going to be translated into a protein. So then this is your this is the protein that's been expressed from your paternal allele, and this is the protein that's been expressed from your maternal allele. So in a normal cell, you're going to have proteins that have been expressed from the paternal allele and proteins that have been expressed from the maternal allele, and that cell is going to be healthy, okay? But what can happen is that you can have a mutation in one of these alleles. So if there is a mutation, this may result in, so what can happen is that maybe the DNA gets, maybe the promoter region gets mutated, so then the binding site for RNA polymerase is mutated, then RNA polymerase can't bind, then you won't have any transcription occurring. If you don't have any transcription occurring, then you won't get translation, and you won't get, so if this occurred in the paternal allele, then you would get no translation of the paternal protein. Or you could have a mutation in the DNA that doesn't affect the transcription, but maybe it changes the amino acid sequence, and so then your protein becomes abnormally folded, so then it's non-functional. Okay, so in either case, the protein is now, you've lost that function, so it's a loss of function mutation. And it means that there's only now half the amount of functional protein inside the cell. And so if the amount of this protein was not enough for normal functioning, then you will get the disease occurring, okay? So I'm just going to draw up a few scenarios here. So now we've had the normal cell, and then we'll have, let's say, we'll have a cell with a recessive mutation. So a cell with a recessive mutation. In this case, it's heterozygous. Okay. So we've got, it's heterozygous, so only the, let's say the paternal allele is mutated. And so then we have, let's say the paternal protein is non-functional and it gets degraded. So then now the paternal protein is non-functional, and we only have the maternal protein inside the cell, so there's only half the amount of protein. But if it's a recessive disorder, what that means is that half the amount of protein is enough for the normal functioning of the cell. So the disorder that you guys learned about was phenylketonuria, or PKU. So that's a recessive disorder, which means that if you're heterozygous and you only have one allele that's knocked out, half the amount of protein is enough, okay? And that's why it's a recessive mutation. This person is going to be fine, okay? But if this person, if another person, and this person is now homozygous, recessive, so both of their alleles have the mutation, and now both the paternal allele and the maternal allele is mutated, which means that they now have no functional protein inside that cell. Then this person will now have the disease phenotype, or if this was the enzyme, if this was the gene for PKU, then this person would have PKU. All right. Now, let's say let's now say that this was a let's now say that this is a dominant mutation. So again, it's going to be heterozygous. So this person only has one allele that is mutated, and so they have half the amount of functional protein inside the cell. But if it's a dominant disorder, half the amount of protein is not enough for normal functioning. And so you'll have the disease phenotype occurring. And so the example of a dominant disease that you were told about is maturity onset diabetes of the young, or MODY2. So this is where there's a mutation in the gene for glucokinase. So if this was the gene for glucokinase and one of the alleles was mutated, and they only had half of the amount of functional protein, that wouldn't be enough for normal functioning, and so they would have MODY2. And the function of glucokinase, it's an enzyme that phosphorylates glucose, and it's essential for monitoring the blood glucose levels. Okay, and since these people can't monitor the blood glucose levels, they're going to have they're not going to produce enough insulin, and so they'll have high blood sugar levels. It's called maturity onset diabetes of the young because it looks like the diabetes that you get when you're older. So it looks like type 2 diabetes, but because it's a genetic disorder, you get it when you're young. So it's diabetes that young people get, but it looks like the diabetes that old people get. All right. So then we'll talk about phenylketonuria. So phenylalanine is, so who can tell me whether phenylalanine is a polar or a non-polar amino acid? Yep, so it's non-polar. So if you look at the picture on the right, it's just got a benzene ring on it, and there's no oxygen in its side chain, so it's going to be non-polar. What about tyrosine? Yep, so tyrosine's polar because it's got an oxygen in it. It's got that OH or hydroxyl group, which means it's going to be polar. And if it's polar, it's going to be hydrophilic. So phenylalanine is a non-polar amino acid, so it's going to be present in the foods that you eat that contain protein. And it's broken down into tyrosine. So you've got phenylalanine, which is broken down into tyrosine, and the enzyme that carries this out is called phenylalanine hydroxylase. So if you look at the name, it's an enzyme that adds an hydroxyl group to the phenylalanine, right? So if you look at the structure of phenylalanine and compare it to tyrosine, you can see that tyrosine's just got one hydroxyl group on it, and that's what this enzyme is doing. So for both alleles of the gene that encodes phenylalanine hydroxylase have to be mutated in order for an individual to have PKU, like I talked about before. because half of the amount of protein is enough for normal functioning. So it's a recessive disorder. Okay. But that being said, that also kind of depends on the environment. Okay, so if you're eating a normal diet and you have, so let's say you're heterozygous for this mutation and you have half the amount of protein, right? So if you're eating a normal diet, you'll be fine because half the amount of this enzyme is enough to deal with the amount of phenylalanine that you're eating. But let's say that you're on a really high protein diet and you are consuming lots and lots of phenylalanine, then you might be consuming too much phenylalanine for half the amount of protein to deal with. So then in that case, you would show the signs of PKU. So whether it's recessive or dominant is also going to be affected by environment as well. But that's a bit more complicated. I just wanted to make that point. But for normal people, it is a recessive disorder. Okay. So if you knock out this, if both alleles are mutated and you have no functional phenylalanine hydroxylase, then you're going to have an accumulation of this phenylalanine amino acid, and it's going to be converted into it's going to be converted into other chemicals which are harmful, and that's going to cause brain damage and other things. And the specific mutation is a base pair substitution. So you're going from, if you look at that, at the diagram at the bottom of the page, you can see that the DNA goes from a C to a T. Okay, so that's a base pair substitution if you're just changing it, and that results in the codon being changed from a CGG to a TGG, and the mRNA codon, which is at the bottom there, is going to be changed from CGG to UGG, and that's because RNA uses uracil instead of the thymine. Okay. And as a result of that DNA base change, it's going to change the 408th amino acid from a positively charged arginine to a non-polar tryptophan. Okay, so if you look at that, the structure of tryptophan in that diagram in the top right-hand corner, that's a non-polar amino acid. And if you can imagine that, let's say you had, so let's say that this here is where the normal, so this is the normal protein and it's got an arginine side chain here, and arginine is a basic amino acid. Okay, it's a polar basic. So if it's basic, it's going to accept a proton and it's going to be positively charged. Okay, and if it's positively charged, well, it's going to be polar, so it's going to be found on the surface. But then if you, now this is the mutated protein. So if you change that side chain to a non-polar tryptophan, this is non-polar, so it doesn't like to be exposed to the aqueous environment. And so it's going to result in the shape of the protein changing. So it's going to have different folding. So for example, it might look like this to try and get away from that aqueous environment. This isn't like, we know that it goes from arginine to tryptophan, but this is like just an example of how the folding might change. This is not exactly how the folding changes. Okay, so this is just an example. Or it could be, the other way it could be is that, let's say, maybe the arginine is on the inside. So maybe the arginine is here and it's positively charged, right? And then maybe you have a amino acid side chain on this side that's negatively charged. And if you've got a negative charge and a positive charge, there's going to be an electrostatic interaction between them, right? And that's going to stabilize this, the fold in the protein. But let's say if it got mutated to a tryptophan which isn't charged, and you've still got the negative charge here, now there's no electrostatic interaction which is stabilizing that fold. So then this protein might just unfold, and now it's got an abnormal folding. And that's going to affect the function of that protein. Okay. Yeah, I just use this as an example. That's not actually, I don't actually know how the arginine changing to tryptophan actually affects the protein. I just know that we just know that it gets abnormally folded. Okay. So because the change in the DNA resulted in a change in the amino acid sequence, this is an example of a missense mutation. Okay. So does everyone know what a nonsense mutation is? What's a nonsense mutation? Yeah, so if you have a nonsense mutation, the change in the DNA sequence results in a stop codon, and the protein is stopped prematurely. So you're going to have a shorter protein, and that's going to be non-functional. And there's also something called a silent mutation. So what's a silent mutation? Yeah. So that's when the change in the DNA results in a different codon, but it's still the same amino acid. Okay. So there's no change in the amino acid sequence. So at the bottom there, you've got the effect of the environment. Even if a person has 2 mutated alleles and they don't have any functional phenylalanine hydroxylase, they can prevent the phenotype from occurring by avoiding phenylalanine in the diet. And also you have the effect of genetic variation. So the other genes in your body is going to affect the severity of your PKU. So the efficiency of the phenylalanine transporters at the blood-brain barrier is different depending on which allele of the gene you have. Okay, so if you have a very efficient phenylalanine transporter, you'll get a lot of phenylalanine into the brain, and that's going to cause a lot of damage. And vice versa. So for these genetic disorders, you can perform genetic testing, and So what will usually happen is that you'll want to first of all obtain some cells from that person. So usually that's going to be a swab on the inside of the mouth, and then you'll get some epithelial cells from the inside of the mouth. And then you'll try and extract the DNA from those cells. But you've only obtained a few cells, so those cells don't contain enough DNA in order for you to do all your analyses on it. So in order for you to analyze that DNA, you need to have a lot of it, especially of just that portion of the DNA. Remember that you have 6 billion base pairs in your DNA, and the gene that you're trying to analyze is only a small part of that. Okay, so you need to get many more copies of that small area of interest. And how are we going to do that? Yep, so PCR. So you're going to run the polymerase chain reaction. And that's going to require the use of a primer that's specific for the sequence that you want to analyze. So I won't go over PCR here because you guys probably all covered that in cells 191, but just make sure that you guys understand the steps required in PCR and what's the ingredients as well. And so once you've used PCR to amplify that DNA, then you're going to use a restriction enzyme. So you're going to mix the DNA with a restriction enzyme that recognizes the mutation. Okay, so what does a restriction enzyme do? What will it do to your DNA? Yep, it'll cut it. Yeah, so if you have the mutation, so in that picture on the right, you can see that it has the mutation. So the restriction enzyme, which in this case is called HIND3, that's going to cut at that site, and it's going to result in 2 smaller DNA fragments. Whereas if you don't have the mutation, so if you have the wild type sequence, so wild type just means normal, it's not going to recognize that that gene, and it's not going to cut it. And so if the mutation is present, then the DNA will be cut into 2 smaller fragments, whereas the wild type will not be cut by the restriction enzyme. And then the 2 smaller fragments can be detected by gel electrophoresis. Okay, so I'll spell that out, gel electrophoresis. And so if you have 2 smaller fragments, you'll get that, you'll get two distinct bands, whereas if you don't have the mutation, then you're just going to have 1 band of DNA. Okay, and so this is just one method of DNA testing or genetic testing. There's lots of other ways that you can do genetic testing, but this is the specific example that you need to know about. Now we'll cover cancer, and then we'll have a quick break. So cancer is due to cells that have acquired mutations that allow them to grow and divide uncontrollably, and are unresponsive to signals that would normally stop them from dividing. So what that basically tells you is that cancer can arise from any cell in your body. So it's a bit stupid when you look on Facebook and you see videos that say like, oh, we found the cure for cancer, or like there's a cure for cancer, but like it's being hidden, because like cancer isn't just one disease. It literally can arise from any cell in your body. So there's so many different types of cancer, and the only thing they have in common is the fact that they're growing and dividing uncontrollably. So it's a very difficult thing to treat. So like, it's just a bit stupid if when you see those kinds of posts or like things. So tumor suppressor genes encode proteins that normally inhibit cell growth. Okay, so remember that a tumor is made up of cells that are growing and dividing. So if you want to suppress a tumor, you'd want to inhibit cell growth, right, and cell division. And for example, the retinoblastoma gene, or the RB gene, that encodes the retinoblastoma protein, so that inhibits cell division. Okay. And that, so usually require both copies or both alleles of the tumor suppressor gene to be mutated for regulation to be lost, hence they're recessive. So what that means is that even if you lose one copy of a tumor suppressor gene, usually that's going to be enough for it to inhibit cell division. Okay, so that's why it's recessive. In order for you to lose regulation or inhibition of cell growth, you need to acquire mutations in both copies of the gene. And then in contrast, proto-oncogenes encode proteins that normally encourage the cell to grow and divide. So if the gene gets mutated and results in increased activity of the gene, then it's now an oncogene, because the protein now encourages tumor development. So onco means tumors, so an oncogene, and because the oncogene is now far more active, you only need one copy to be mutated in order for, and so it's a dominant. So proto-oncogenes are dominant. Mutation, so proto-oncogene mutation, it could either cause increased transcription, and the mechanism of that could be maybe the binding site in the regulatory region that binds the repressor, transcription repressors, has now been mutated, so now the repressors can't bind. Or you could have a mutation which means that the protein has increased activity. So for example, it could, maybe there's a mutation in the binding site for inhibitors, and if the inhibitors can no longer bind, then it's going to be more active. Okay, and the disease, the specific cancer that you were told about is chronic myeloid leukemia, which occurs due to a translocation event. So translocation means that you've moved one part of the chromosome to another chromosome. And so when that occurs, you've got basically two genes that are normally separate becoming one gene. So the BCR and the ABL gene are combined, and then you form a new gene, which is called BCR-ABL, and that is an oncogene because now it's this new protein or this new gene now creates a protein that promotes cell division. Okay, and in order for cancer to occur, so this is in general, you have to have loss of tumor suppressor activity, which means that you have to mutate both copies, and you have to gain an oncogene mutation. So then how many mutations would you require as a minimum? Yep, 3. So you're going to require 3 mutations. So you'd have to have 2 mutations in a tumor suppressor gene, and you'd have to have just 1 mutation in an oncogene. Okay. And the reason for that is that if you just lost, let's say you had mutations in both of your tumor suppressor genes, but you also didn't have a mutation in your oncogene, then there'd be nothing to inhibit, so you'd be fine. So even though you've lost your ability to suppress a tumor, there's nothing driving the division of those cells, so there's nothing to inhibit. And the other way around, even if let's say your tumor suppressor activity was normal, and you've gained an oncogene, even though you've got something encouraging like cell division and cell growth, you've got that tumor suppressor gene which is acting to stop that division. So in that case, you're also safe. So that's just a mechanism of how cancer is still like quite, you don't really get cancer until you're really old and you've acquired a lot of mutations. So that's just how the body protects itself from cancer. So you need to have all 3 mutations occurring. And the chances of all 3 mutations is extremely low, but because there's such a large number of cells in the human body, 37 trillion cells, you can still get cancer, and it's still relatively common. And some people can inherit a mutated allele in a tumor suppressor gene. So this means that they only need to develop 2 extra mutations in order for cancer to occur. So that's like playing lotto, and so if you want to win lotto, you have to get 6 numbers correct, right? But let's say you always know that you get 1 number correct, so then your chances of winning lotto are going to be a lot higher, right? So an example of one of those is BRCA1 and BRCA2. So you guys might have heard of that because Angelina Jolie had that mutation. So BRCA1 is like a tumor suppressor gene, and people that inherit a mutation in one of them have a much higher chance of developing breast cancer. So what Angelina Jolie did, because she had genetic testing done and found out she had the mutation, she got both her breasts cut off, which is called a double mastectomy. And so on the last page, you've got 1 sentence. So cancer treatment includes inhibition of the enzymes that are necessary for cancer cell growth and division. So that's the end of Tute 6, and we'll have a quick 5-minute break and then start again at 8 o'clock.